The nano world refers to the realm of nanotechnology, which involves manipulating and studying materials at the nanoscale, typically at dimensions smaller than 100 nanometers. At this scale, unique properties and behaviors emerge, enabling scientists and engineers to design and create new materials with enhanced characteristics for various applications from medicine to electronics. Nanomaterials are substances engineered or manipulated at the nanoscale, typically ranging from 1 to 100 nanometers. At this size, materials exhibit novel properties due to quantum effects and increased surface-to-volume ratio. Nanomaterials can be organic, inorganic, or hybrid structures, offering unique functionalities like enhanced strength, conductivity, or reactivity. Applications span diverse fields, including medicine, electronics, energy, and manufacturing, harnessing these materials' exceptional properties for improved performance in innovative solutions. Nanoscience is the study of phenomena and manipulation of materials at the nanoscale, typically ranging from 1 to 100 nanometers. It explores how properties change at this scale and aims to understand and utilize these changes for various purposes. Nanoscience encompasses physics, chemistry, biology, and engineering, playing a crucial role in the development of nanotechnology. Researchers in nanoscience work to unravel the unique behaviors of materials at the nanoscale and apply this knowledge to create innovative technologies with applications across diverse fields. Nanotechnology leverages unique properties emerging at this level to design and create novel structures and systems. Nanotechnology spans various disciplines such as physics, chemistry, biology, and engineering, and is employed in diverse fields like medicine, electronics, energy, and material science. Its potential lies in the ability to engineer materials with precision, enabling the development of advanced technologies and solutions for a wide range of applications.
Good day everyone, I'm Rochelle Pilisan and my topic is all about important personages who have contributed to the growth and study of the Nanona work. The first paragraph is at established historical foundation, scientific prestige, the failed idea support among advocates and science. And the second paragraph is recognize best computer science potential at the establishment, anticipate really the implications, advancement need prepare awareness about. And the third paragraph is development by the earning force further by enabling precise at the significantly understanding materials. And the last paragraph is 1980s significant due to two science creation played crucial role. The field, the discovery base, unique, subsequently understanding, structure, a few expanded ability, manipulate materials at the groundwork, further development.
So, initial commercial application, the early 20s saw the beginning of the use of nanotechnology in commercial product, although most of applications are limited to the block or use of passive nanomaterials. Examples include titanium dioxide and zinc oxide. Nanoparticles in sunscreen, cosmetic, and some food products. Silver nanoparticles in food packaging, clothing, disinfectant, and household application, such as silver nano, carbon nanotubes for stain-resistant textile, and zirconium oxide as fuel catalyst. As of March 10, 2011, the project on emerging nanotechnologies estimated that over 1,800 at 100, manufacturer identify nanotech products are obviously available, with new ones hitting the market at a pace of 3, 3 to 4 per week. Potential uses. The Joint Center of Biotechs in Canada ranked potential use of nanoscience and nanotechnology with respect of development. The top 10 use follow BBS News 2005, energy production, conservation, and storage, enhancement of agriculture productivity, treatment and remediation of water, screening and diagnosis disease, system for drug delivery, processing and storing food, controlling air pollution, construction, monitoring health, detecting and controlling pests.
So, what is gene therapy? Gene therapy is an experimental technique that uses genes to treat or prevent disease. In the future, this technique may allow the doctors to treat a disorder by inserting a gene into a patient's cells instead of using drugs or surgery, because some diseases are caused by errors or mutation of specific genes. Gene therapy delivers DNA into cells to replace mutated or missing genes or to add good genes. So, how does it work? Gene therapy is designed to introduce a genetic material into cells that compensate for abnormal genes. Here, the patient is being given a healthy version of gene that makes the cell functional and restored. As you can see on the figure, a 9-year-old boy, Creed, who has a genetic form of blindness, is one of the first people in the United States to receive a recently FDA-approved gene therapy for a congenital form of blindness called Leber congenital amaurosis. The treatment is an injection of virus delivered through a hair-thin needle underneath a patient's retina. Within a month of gene therapy, Creed was able to see details of the world we'd never seen before. So, let's now move on to the two types of gene therapy. Virtually, all cells in the human body contain genes, making them potential targets for gene therapy. However, these cells can be divided into major categories. The somatic cells, which is most of the cells of the body and cells of the germline eggs or sperms. In theory, it's possible to transform their somatic cell or germ cell. There are two different types of gene therapy, depending on which type of cells are treated. So the first one is the germline therapy. Involves the modification of the genes inside germ cells, sperm, or ova. Unlike the somatic gene therapy, only involves the insertion of therapeutic DNA into body cells and not the germ cells or gametes. This means any effect of the therapy are confined to the individual being treated in or not inherited by future offspring. In summary, the distinctions is that results of any somatic gene therapy are restricted to the actual patients in or not passed on to his or her children. All the gene therapy to date on humans has been directed at somatic cells, whereas germline engineering humans to the main controversial and prohibited in or instance the European Union. So let's now move on to the two types of somatic gene therapy. In vivo gene therapy means that gene therapy is administered directly into the patient. The targeted cells remain in the body of the patient, so the gene is being transferred to a patient cells while still in the patient. However, a gene cannot be introduced directly into a patient's cells. Instead, a vector, which refers to a virus, is genetically engineered to transport the gene. So the next type of somatic gene therapy is the ex vivo therapy, gene therapy. In ex vivo gene therapy, the targeted cells are removed from the patient, and gene therapy are administered cells and vector before they are returned to the patient's body. Herd of gene is being transferred into the cells in laboratory and then injected back into the patient via IV directly a specific tissue. So, as you can see in the figure, in ex vivo gene therapy, the cells are being taken out from the patient and these cells will be modified in the laboratory. When the cells become restored or functional, the patients will be now returned at the patient's body.
So, good morning everyone. I'm Crazy Mipahit. I'm going to report the three approaches to gene therapy. So, first is gene augmentation therapy. So, this therapy is like fixing a blueprint in a house. Imagine if there's a mistake in the plan and a room doesn't work right. So the plan serves as your gene and the room as your plan. So with this therapy, scientists can bring in a correct plan or even add a new design to make that room function properly. So this is like giving your cells the right instructions to build things the way they should be, and this helps fix problems caused by mistakes in our genetic instructions. So next is gene inhibition therapy. So this therapy is like turning down the volume on a too loud radio. Some genes in our body make things we don't want, like too much of a protein causing trouble. So in this therapy, scientists find a way to make those genes quieter, like putting a mute button on them. So this helps to control the production of problematic stuff in our cells, making sure they don't cause as much trouble. So lastly is the killing of specific cells. So when it comes to targeted cell killing, it's a bit like weeding a garden. If there are some bad plants or problematic cells, we want to get rid of them without harming the good ones. So with this therapy, scientists figure out how to remove the specific bad cells, like putting out weeds from a garden, leaving the healthy ones behind. So this is all about precision, fixing only what's broken while keeping everything else in good shape.
Good afternoon, ma'am. My name is Valerie Shin Pivot, and this is my topic, the advantage and disadvantage of gene therapy. So, what is gene therapy method? Gene therapy is a technique that modify the person's genes to treat or cure disease. Hold promise to treat many diseases, but there are still new approaches to treatment and may have potential risk could include certain type of cancer, allergy reaction, or damage to the organ. Sa advantage, kaya example diri na give hope of healthy life for cancer patient. It have made genetic therapy much safer. In the future, genetic may be used to prevent, treat, or cure certain inherited disorder, and also may be used to treat cancer or infection, including HIV. Genetic therapy can be a life-saving for some people with a specific medical condition. And sa advantage ay sa disadvantage lang namo sa gene therapy is sa example na guide the genetic testing, screening, and research in finding availability of certain type of certain gene is very controversy. So, sa genetic therapy hold promise to treat many diseases, but there are still new approaches to treatment and may have potential risk could include certain type of cancer, allergy reaction, or damage to the organ. Sa gene therapy method, dyan na si Zakay, dabo ang mahitabang sa patient, pero pero makakos si zanan side effect. How kan sa possibility of causing a tumor or higher risk of increasing allergy. And if delivering a gene to the wrong tissues would be in a infection and it could cause health problem for patient. If mali ang imo pag-deliver ng gene sa patient, it would cause some infection, or it could cause of health problem and also have risk of increasing allergy or shorted life for patient. And if mali ang pag-deliver ng gene sa patient, it would cause some infection or it could cause some health problem and also risk of increasing allergy or shorted life for patient. Kaya imbis kay ala papadugazon ang kinaboy sa patient, mas madali-dali noon because of wrong sa pag-deliver ng gene sa patient. So gene therapy can be safe, but it also carries potential risk like any other medical procedure. How that sa example kay ina na makakause sa nan cancer or infection sa nan makakause ng tumor. So sa gene therapy medyo dili gyapon siya safe. So that's all ma'am, thank you.